Hi, in this presentation we are covering electromagnetic receptors. Our objectives are to define electromagnetic receptors and photoreceptors, describe the basic structure of the human eye, differentiate between rods and cones, and explain how the photoreceptor cell detects changes in light. There are a variety of electromagnetic receptors. They detect a wide range of electromagnetic radiation. Some receptors detect UV light, others visible light. There are receptors for infrared or electrical energy or magnetism. It depends on organism what type of receptor they have. Humans, like other vertebrates and a lot of different animals, have vision. Vision involves photoreceptors. Photoreceptors are electromagnetic receptors that respond to visible light. Sharks, rays, and some fish have eyes. They have photoreceptors for vision, but they can also detect weak electrical fields with other electromagnetic receptors. If you remember from our shark lecture on the ampullae of Lorenzini. Some types of snakes, called pit vipers, have infrared receptors. They're located in sensory pits so that they can detect the infrared radiation given off by living organisms. And some types of birds, which can navigate relative to Earth's magnetic field, have electromagnetic receptors. We're going to focus on photoreceptors. This is an adaptation that is very ancient. It's shared by nearly all animals and a lot of other organisms, including protists. So the simplest organs for photoreception are eye spots. And we can find eye spots in a lot of protists and even some invertebrates like jellyfish. Eye cups are a little more complicated, and this diagram right here shows an eye cup in a flatworm or a planarium. So our flatworm. Notice the basic structure. We have pigmented cells that protect the photoreceptors. Here's our photoreceptors. Pigmented cells protect the photoreceptors from light coming in from this direction. Light coming in from this direction will stimulate the photoreceptors to send a message to the brain. These are good for responding to the presence, or absence of light, but they're not good for image forming. Compare that to the compound eyes found in many arthropods and some annelids. We'll look at a picture of that next. And the single lens eye found in vertebrates and some invertebrates. So the compound eye, which is found in arthropods and some annelids, this is actually image forming. And it consists of multiple light detectors that are called ommatidia. So here's a diagram of one ommatidium. You see a clear membrane, a little crystal cone for a lens, and then here are our photoreceptor cells. An invertebrate eye, compound eye, will consist of many ommatidia. Depending on species, it can be hundreds of ommatidia or even more than 10,000 ommatidia. The ommatidia will detect intensity of light and also color. That makes a compound eye excellent for motion, but when you compare it to the single lens eye, even though it's forming an image, it doesn't get the sharp image resolution that a single lens eye will get. Humans and other vertebrates, as well as invertebrates like some of the mollusks, octopods and squid, have a single lens eye. So we're going to take a moment to look at this diagram and note the structure. I want you to note this outer membrane. This is called the sclera. This is what we call the whites of your eye. At the front of the eye, the sclera becomes clear, and now we call it the cornea. So when light enters the eye, it travels through the cornea, then it travels through a hole called the pupil that is in a circular muscle called the iris. And in our eye, it's the iris that's colored. So if we talk about somebody's beautiful brown eyes or beautiful blue eyes, we're talking about their iris. Light travels through the pupil, and then through the lens here. The lens is another clear structure, and its job is to focus light on this back tissue lining the back of our eye here, which is called the retina. And the retina is where we have our photoreceptors, concentrated in the fovea, which is our focal point, and not in the optic disc, which is where the optic nerve attaches to the back of the eye. Look at another feature of the lens. The lens changes shape in order to help us focus. So for near vision, the lens gets rounder. Muscles contract to make the lens rounder, and that focuses light so it's covering a broader area on the retina. When we're doing distant vision and we don't need that sharpness of vision, the muscles relax, the lens flattens, and we're focusing light on a narrower region of the retina. What happens when people get older, they start needing these glasses, is that our lens gets less flexible. It doesn't change shape as well for that near vision. In the retina are the photoreceptors, and I'm again focusing on the single lens eye. Photoreceptors include the rods, and so here's a diagram of rods. Notice it's a living cell. So here's the cell with the nucleus. Here are extensions that will secrete neurotransmitters to talk with neurons. And then back here are membranous discs that have a protein pigment called rhodopsin. And rhodopsin is very sensitive even to dim light. When light hits this protein, it changes shape. When enough of these proteins change shape, that stimulates the rod cell to send a message to the nearby neurons. In your retina, in the human retina, we have approximately 120 million rod cells. And again, they're very sensitive to dim light, but they're not good for color vision because there's only one type of pigment. Compare that with the cones, and these are our cones. Cones, there are different pigments. They're similar to rhodopsin, but they respond to different light ranges, different colors. So we're going to call them photopsins here. Humans have 3. Different vertebrates may have more than 3. I'm jealous. And in our retina, we have about 6 million. They're all located in our focal point, which is the fovea. A cone is not as light sensitive, so they don't respond well in dim light levels. It's why at dusk, you don't see colors as well as you see shapes. And for humans, we have 3. One responds better to red, one responds better to green, one responds better to blue. And that just amazes me. You know, I go to paint stores and I look at all the different paint chips, and it amazes me how many different colors that we can distinguish. We can distinguish all those colors and yet only have 3 different types of cones. So our blue cones, which are absorbing in this range, and our green cones in this range, and our red cones more in this range. So it is the pattern of cones that are stimulated that our brain interprets as different colors. It's mind-boggling. How do the photoreceptors work, though? You see a diagram of our rods. When they're not detecting a stimulus, you know, it's dark, no light, absence of light, the membrane is actually depolarized. Now remember back to learning about action potentials. Depolarization is what happens when we're sending an action potential. And that's what's happening here. We've got a neurotransmitter, which is glutamate, and the rods are constantly sending little bits of that neurotransmitter and communicating with the neuron. So even though it's dark, we have communication. So what happens when it's light? So when light is present, the membrane is hyperpolarized. Remember, hyperpolarized means that it's not sending that signal. We've closed our sodium gates. We're reestablishing that membrane potential. We're not sending an action potential. So we're actually not sending that neurotransmitter. Well, it's the absence of the neurotransmitter that signals that neuron to send a message to our brain. So photoreceptors are kind of backwards, but it gives them a very rapid response time. For the structure of our retina, again, the fovea is our densest, right here. It's our densest region of cones. Highest visual acuity. When we're reading, we're focusing an image on our fovea. As we go off to the periphery of the retina, cones become less common, rods more common. That's our peripheral vision. And then our optic disc right here is our blind spot because there are no rods or cones. Another thing that amazes me, this is the back of the eye. We have our pigmented layer. Here are the photoreceptors. Behind the neurons that the photoreceptors are talking to, and more neurons, and a whole network of neurons heading for the optic nerve. So light is actually traveling through multiple layers to get to our photoreceptors. To summarize, let's do a couple questions. Just make sure it's all clear. What structure of the eye focuses light on the retina? That would be our lens. And which region of the retina contains only cones and is the sharpest area of visual acuity? That's our fovea, our focal point. The big ideas for this is that electromagnetic receptors are designed to detect changes in electromagnetic radiation, including visible light, UV light, magnetism, electrical fields, and infrared. Photoreceptors refer to a specific type of electromagnetic receptor that respond to visible light. And photoreceptors are an ancient adaptation found in protists and many different animals. I'll see you in our next presentation, which is our case study.